We get a chance to hear from Jon Bakker of the University of Washington and hear about how we can successfully adapt to changing conditions across these broad and varied landscapes and habitats. Jon's a professor of ecology at the University of Washington and has studied restoration in prairies, forests, sagebrush steppe, wetlands, and subalpine meadows. In this presentation, he'll describe two complementary approaches for restoration projects: staged scale restoration and distributed experiments. Staged scale restoration is a strategy by which treatments are tested experimentally and then adapted to address the unique goals and challenges of individual sites. Efforts start small and over time are scaled up while being refined to meet the needs of that site. Distributed experiments are simple experiments replicated at multiple sites. The power of this approach is the opportunity to synthesize patterns across sites. Coordination of efforts, data sharing, and analysis is key and could be relatively inexpensive if centralized by an academic partner. So looking forward to hearing from Jon how to implement the adaptive riparian restoration in a coordinated approach.
So I didn't realize Adam was going to give the whole summary there, but I'll just try to illustrate those ideas through some of these, through what I'm talking about here today. I'm going to start with a couple of assumptions. The first is that restoration is dynamic. That's one of the things that makes it exciting, but we'll work on different sites and we have lots of different situations that we encounter when we're doing restoration in those different projects. And so every site is often unique in some way that we want to recognize. The second assumption is that experimentation is the best way to improve restoration. So learning from our mistakes or just trying different things and seeing what works is one of the ways to do restoration and to improve the ways that we do restoration, both in terms of efficiency and also in terms of the effectiveness of the restoration itself. But not everybody wants to be an experimental designer or wants to design an experiment or think about things in sort of an experimental framework. Not everybody is interested in organizing or analyzing data. So there are some other, those are my other assumptions that I'm making here. And so I want to talk about these two ideas and how they can be put together, perhaps uniquely in the context of riparian restoration and the opportunities that that might offer for riparian restoration. So coordinated distributed experiments. One of the factors that led to this was thinking about how can we identify broad patterns. We're trying to understand how systems work. If we're doing restoration, how can we improve our understanding of when and where particular methods are appropriate so that we can improve the kinds of restoration that we're doing there. And so these coordinated distributed experiments have been proposed that they have potential global applicability, right? If we're trying to understand things on a really large scale, and I'll show you some examples that are done on really large scales, but they don't have to be. And I'll also show you some examples that are done on more regional scales to improve our understanding of what's happening in ecological systems. So let's walk through each of those components of this term. It's a little bit of this phrase, coordinated distributed experiments. So when we say things are coordinated, what we mean is that there are common protocols that are being applied. The data are being curated or collected and maintained in a single location. There's analysis that happens across multiple, across all the different sites that are involved. So we have this coordination in terms of the initial ideas that are being applied, and also so on the beginning of a project, but also in what we might think of at the end of the project in terms of the organization and analysis of data, preparation of reports and figures, and all of those kinds of things can all be coordinated in that regard. Distributed is referring to the fact that we do this at multiple sites. Often, and perhaps ideally, those are sites that span different jurisdictions. They might be on different kinds of rivers. They might be in different kinds of land use histories. All of those kinds of all of that variation can be incorporated into the sites that are selected. And then experiments. And what that means is that we're not just doing one thing at any particular site, but we're always thinking about this in an experimental framework. So we have some treatments we're interested in. Maybe we're going to be planting at 2 different densities, or we're going to be planting 2 different kinds of stock types, or we are doing different site preparation treatments. And we're not necessarily doing all of those things. Maybe at a particular site, we might say, okay, well, what happens if we use our standard method and we sow or we plant at a higher density, right? And we have areas that we do each of those. And then while we're doing that, we're also incorporating some of the key experimental ideas related to randomization and replication. So we're randomly choosing which areas get which treatments. We're using replication, so we have not just one area that's planted at one density and another area that's planted at a higher density, but maybe we have those scattered across a landscape with more representation that way. So I'll show you 4 examples of this. The first one is one I've been involved with. The others are ones that I'm aware of, but I'm not a member of. So the nutrient network, or NUTNet as it's commonly abbreviated, is a global research cooperative, or a global coordinated distributed experiment that's been around since 2007. In the realm of plant ecology, this has been a really influential group in terms of the kinds of products that have been produced from it. And I've got an image of one of the papers. I've got several images of different papers here, and I'll just pause and say, if you want to see any of these, send me an email and I'm happy to share PDFs with you for them. So nutrient network is set up to ask some basic questions in plant ecology. And what they're specifically interested in are what happens to grasslands when we add fertilizers to them, and what happens to grasslands if they aren't grazed by mammalian herbivores. And so we have an experimental design that is testing this. We have these different plots shown by this little square here. There's a series of plots, and then some of them receive no fertilizers, some of them receive nitrogen, some receive phosphorus, some receive potassium, and then different combinations of those. So we go out once a year, we add nitrogen to the plots that need nitrogen, we add phosphorus to the ones that need phosphorus, et cetera. And then we just stand back and observe what happens in those plots. And then we have some fences on a few of these plots. So we have some of these, you can see some fences in the image up above here. Some of these areas that are fenced don't have any fertilizers, and some of them are fenced and are receiving all of the fertilizers. And so we can look at, well, what is the effect of these nutrients, and what is the effect of the fences, and understand how these systems are interacting. That design was set up, and then there was basically an open invitation for ecologists around the world to participate in that. So if you want to participate, here's the protocol for how to do this experiment, do it at your site, share your data with a central kind of headquarters, and in return, you get access to all of the data from all of the sites that are involved in this. And so the dots on this global map are the sites that are involved. The color coding is reflecting whether they are just doing the nutrients, or they're just doing the fencing, or some other variations like that. But you can see the representation all around the globe, which has been one of the really interesting aspects of this in terms of trying to understand grasslands as a system. Right? We think about grasslands as many people think about grasslands as relatively simple systems. They're hugely important around the globe. They're about 1/3 of the global land mass. And there's a lot of variety within grasslands themselves. So we use these data to look at things like, how does the productivity, how much plant material grows in a particular site? How does that relate to how many plant species we find in the site? And this was one of our first papers that came out on this topic, and the take-home message is in the title. If you know something about how productive the site is, you really don't know anything about how many species that particular site would contain. what gets more interesting, and this is a pretty busy graph, and I'm not intending you to be able to read all of this in any way. Each of these little boxes here is one of the sites that was included in that analysis. And so what we're looking at here is that relationship of productivity and richness for individual sites. There are some of these sites that have lines in them. That means that there is some sort of statistical relationship. But there's a whole bunch of them that don't have any lines in there at all. So there's no relationship amongst those variables in those particular sites. And where there are relationships, sometimes they're positive, sometimes they're negative, sometimes they're convex or concave. So there's a lot of complexity in those relationships. So we have a global pattern, but we have a lot of variation among individual sites as well. The site that I have highlighted in red there is the site that I maintain. This is Smith Prairie on Whidbey Island. And that's a site that I've been doing these fertilization treatments on every year since 2008. So that's one example of the kinds of questions that we've asked. Here's another paper that we wrote a few years later. So this first paper that we did here, this was based only on what's called our pre-treatment data. So we were just looking at, well, what is the actual productivity and the actual richness on these sites themselves before we do any of these experimental treatments. Once we start to do the treatments, we start to add these different nutrients, then we can start to look at how things like the productivity, how much biomass the plots have, how that changes as a function of fertilizers. In this graph here, these colors are corresponding to the different treatments that have been applied. And the ones at the very top here, so this is after 3 years of treatment. The largest difference here is the plots that receive nitrogen and phosphorus together or receive nitrogen, phosphorus, and potassium. So that combination is more important than thinking about than if we just add nitrogen, shown here in the green, or if we just add phosphorus, which is shown in that blue symbol there. So that's an important kind of global pattern. But again, there's complexities among individual sites. At a quarter of the sites that we included in that analysis, these nutrients had no effect on productivity. So didn't matter if we added nitrogen, didn't matter if we added phosphorus, didn't matter if we added potassium. The productivity was limited by, presumably by other things like water or other factors beyond these nutrients. One last example from this is a study that I'm currently working on for these data. Again, every pie chart here is a different site somewhere around the world. My site that I focus on is shown in the red dot there. What I'm looking at here is how variable the composition is. So which species are present in the plots themselves, and I'm characterizing it in terms of whether that variation is due to the kind of the spatial differences among the blocks of plots, or whether it varies among years, shown by the yellow, or whether it's affected by the nutrients, shown by the red and orange colors. And what I think is interesting is just to kind of step back and look at how much variation there is. Some sites have huge variation from year to year. Some sites have huge variation from block to block. Some sites have much larger effects of nutrients than other sites do. So there's a lot of complexity that I think there's more complexity here than we were expecting we would see amongst these responses to treatments. That's thinking about grasslands in sort of in a global perspective, and from using questions that are somewhat that are theoretically interesting but are not necessarily applied questions, right? Most of us, if we're thinking about how we're going to manage these kinds of grasslands, we're not thinking about adding fertilizer to them. We're thinking more about removing or reducing nutrient availability, for example. But there are examples that are doing this in more applied contexts, and that's what I'll show here. This is a project that was led by the Nature Conservancy back around 2010. This was a 5-year project, and it was looking at ways of restoring these grasslands. And what they did in this context was a series of experiments where they did the same experiment at a whole bunch of different sites. And they were trying to figure out which kinds of treatments they needed to apply to the sites in order to improve their restoration success here. Concluded in part that you couldn't do any one thing, and you needed to have combinations of things that you were doing. And also that how you, or the best ways to treat these sites varied among the sites often as well. So you could come up with these treatment combinations, and some treatment combinations were more effective than others. And you get importantly, you get a lot of a better understanding of the region-wide approaches or the consistency of these effects when you think across an entire region like this, as opposed to just doing an experiment at one particular site. So this was called the Collins Project. It was funded by the Collins funding source within the Nature Conservancy, and this ran for about 5 years and is not currently active. Another applied example, the Stand Management Cooperative, is through the University of Washington. And this is a long-term silviculture research project. It's been going for multiple decades now. And it's got a different structure than all of the other ones I'm talking about here, in that it is funded by dues that are paid by members. And so you've got a lot of industry, you've got government agencies involved, and you've got academics involved. And they have the same sort of cooperative or coordinated distributed experimental approach. The dots on the map here are all the different sites where they have their experimental plots set up. And so they'll do experiments in forests across Western Oregon and Western Washington and up into BC. And then follow that same standard approach in all of those different sites and look for patterns in the responses that they see to those treatments. And then the last one here that I want to show is called RestoreNet. This is a group out of Arizona, and they're looking at desert grasslands and ways to restore these grasslands, both by seeding and by trying to improve soil moisture in the plots themselves. And again, they're working in multiple sites that span large gradients in terms of which ecosystems they're in, how much precipitation they receive, how much what elevation they're at, et cetera. And doing the same thing at multiple sites allows you to come up with much more robust conclusions, and conclusions that are more widely applicable than you would ever get if you focused all of your efforts on a single site. So the idea of coordinated distributed experiments has some advantages and some issues. As conceived and as how usually implemented, the focus here is on techniques that are relatively low tech and low cost to implement at any particular site. But then those are leveraged to provide high value because you have that applied in multiple sites, and you can look for those, again, those commonalities or those common patterns across sites. The challenges here include the need to agree ahead of time on your design and methodology so that that gets applied consistently on all the sites. Coordinating efforts so that these things happen in the intended scheduling. And then the ideas of data sharing and analysis. And I know with the nutrient network, the NutNet, the example that I'm involved with, we have at our headquarters, we have a person whose full-time job is to organize the data that gets submitted and then provide it to people who are interested in doing the analysis for that. But that's a little bit of a different context, I guess, because it's working primarily with academics and researchers. And so there's a lot of people interested in using the data. I'd say in the Stand Management Cooperative, for example, they also have a database manager who's organizing this, but then they have a more restricted set of people who are interested in doing the analysis and a much wider range of people who are interested in seeing the results of the analysis. And so they're sharing their research outcomes with the partners who are funding it. All right, so now I want to talk about the second aspect that I want to introduce here, which is this idea of staged scale restoration. And I'll introduce it by way of this question. If you thought just about those coordinated distributed experiments and you're doing the same thing at a whole bunch of different sites, that could limit your ability to really tailor the treatments to the needs of a particular site. And this is something that we have explored in the context of grassland restoration, and we introduced this idea of stage scale restoration as a way to tailor the restoration actions that you take to a particular site. I'll just kind of walk through the schematic here, and then I'll illustrate this with what we've done in the grasslands we were working in. We would start here with this stage one, which is this checkerboard up in the top left corner here, and we would have some treatments that we're interested in testing. They could be anything that we're interested in exploring. And we do that in this experimental array. So we have some areas that receive one treatment, some areas that receive another treatment. In this case, every one of these different patterns would reflect some different treatment, and they're randomly assigned across this landscape, across this array. And so we do that. We allow some time to elapse, and we go back and we walk through this array. We might collect data, we might look at it qualitatively, and we decide which of these treatments seem to be working the best on this particular site. And then once we've decided which of those treatments seem to be working the best on this particular site, then we can apply those on that same site to slightly larger areas. So we're going to, that's the staging here and scaling this up. So in the second stage, in this case here, we take this white treatment, we decide, okay, white treatment seems to be working really well compared to these, the black treatment. So we're going to take the white treatment and we're going to apply it to an area that's 10 times larger than what we started with. And we might do that with a few different treatments from this. We can then step back and observe that, again, how that's working on this particular site. And then we can decide, okay, which of these do we want to now apply even more fully to this particular site? And we can do that in a third stage where we now apply it to an even larger piece of that landscape. So that particular treatment has been customized to the needs of that particular site. We developed this in the context of Western Washington grasslands. We were working with abandoned agricultural fields in this context that look something like the image on the left there. And we're trying to recreate a native dominated grassland that has much more diversity than that abandoned agricultural field, includes some rare plants and rare animals that are able to use it. And so those are our objectives for thinking through this. And so what we tested in this particular context was a variety of site preparation methods. These are methods that we agreed upon in conversation with land managers. So we said, okay, here are the kinds of things we're interested in testing. If these were successful, could you imagine applying this across larger pieces of landscape? And so all of these are things that are operationally feasible to apply on large landscapes. And we applied them to relatively large plots. And the plot size could be tailored for the needs of a particular system. So in this case, we're testing site preparation, and then we're also testing some different seed mixes. So all of this work was done with direct seeding in the fall with a whole bunch of different species, and then observing which of those species we're establishing afterwards. And I think it's helpful to think about this. I walked through kind of a timeline of how that might work, but it's also helpful to think about that spatially. This is our study area at Glacial Heritage Preserve down just south of Olympia. This polygon is the area we're working in, and this is what it looked like in 2006. These white areas are areas that are used for other experiments, and so we're not part of our experiment here. But what I want to just walk through is kind of the process that we went through and how that looks spatially across the landscape. So we started off with that experimental array. So now instead of having those cross-hatching color, I've just used colors to represent different treatments. Each of these represents a treatment. It doesn't matter for our purposes right now what the treatments are. In this particular study, we also actually repeated that experimental array the next year. So we did the treatment, we did the array 1 year. The next year we looked at it, we repeated that experimental array a second time. And then based on what we were seeing in the first year, we thought, oh, the blue and the red colors seem to be working the best. So let's apply those to some larger pieces. But we also had this orange color that has shown up there. What this is our herbiciding treatment ended up being delayed a year from everything else. So we couldn't evaluate it in the very first year. So that showed up in the second year of our study. When we came back and we looked at now both of these arrays, this orange seemed to be functioning pretty well. So we applied that to a larger piece of landscape. And then we also did this experimental array actually a third time here as well, just to understand that. But the red treatment that we had scaled up initially, thought, oh, after looking at it for another year, this doesn't seem to be functioning quite as well as these other ones do. So we won't scale that one up anymore. And then in the next year, we said, okay, this orange definitely seems to be the treatment that is the most effective for our for what we need on this particular site. So we'll apply that to some larger pieces. And then we started to also work on some really large pieces towards the end of the project here. And then in the very last year of the project, it was clear that that orange treatment was the most effective one that we had been working with there. And so we applied that to this much larger piece of the landscape here. So we started with 7 different treatments, and we were able to tailor it to figure out which of these particular treatments worked best on this particular site. We actually did all of that. This was a dissertation project of a student, and he actually did this on each of 4 different sites. We started with the same set of 7 treatments that we were going to test in all 4 sites. By the time we were done and we gotten through the end of that process, the final recommendation for what we were going to do on that site was different for every one of these sites. Even though they're all in Western Washington, some of them are just a couple of miles away from each other, but they differ in terms of which non-native species, which invasive species are problematic in terms of soil conditions, in terms of the weather that they experience. And so we had to tailored our final recommendations for each site based on what we learned by applying all of these initial treatments to each of the sites themselves. So some advantages and issues related to these. We start small and we can adapt to the unique goals and challenges of individual sites. If you only did this on one site, then you would be able to customize it to one particular site, but you wouldn't be able to generalize very well. And so that's why this idea of putting together these coordinated distributed experiments together with this idea of stage scale restoration is, I think, so appealing or potentially appealing for riparian restoration. And what I want to suggest here is imagine what we could do and learn if we were doing all of these restoration projects in a coordinated manner so that we could compare across them instead of thinking about each one as an individual project and its own individual site, or maybe it's an individual experiment, but we don't have any generalizability in that context. So again, just to reiterate, we would the idea here is that we would follow this coordinated distributed experiment where we test the same simple treatments at multiple sites, but we would then also tailor that to the needs of individual sites using this stage scale approach, so that after having tested them initially, we can now start to customize the treatments for individual sites as we scale it up and apply that to larger pieces of a site. There are lots of different ways that this could be done. We could test different things. We could measure different things. How this would be actually implemented in the context of riparian restoration would be an important conversation with practitioners in terms of figuring out what are the key things we want to test, what are the key things we need to measure. But I just put a few examples on the screen there. I think there could be some potential helpful aspects in terms of reduced reporting requirements. If we are able to coordinate the activities and we can produce, for example, one report that spans multiple sites instead of having each individual site having to prepare reports for what happened on their particular sites. And those reports and the analyses would be more powerful because they would be based on a much larger data set than would be possible if individuals were working on one site at a time. And I think it's not insignificant to remember that those reporting requirements could be led by people who like those parts of the project, right? If they like working with data, then give them all the data and let them work on that. And the people who like to be in the field can be in the field tweaking these, figuring out how to improve the effectiveness of these treatments on individual sites. So an example here, this is a study from Oregon State. I put it in here specifically because it is focused on riparian restoration projects. And it's this is actually Lucy Law's thesis defense just from last month. This was highlighted or mentioned in a virtual conference I was at a couple of weeks ago. And given the audience here, I thought some of you might be interested in this as well. They worked in a variety, I think it was 12 project areas throughout the Coos Bay area of Oregon. And they tracked the survival and height growth of 8 different tree species planted in all of these areas, and were able to make much stronger conclusions than they could have if they had just worked in a single site. So conclusion, this is a paper that I think the title is worth remembering in this regard. We're trying to improve restoration outcomes to make restoration more effective and more efficient. And the best way to do that is this idea of having a common experimental design and a common structure that gets applied in multiple restoration projects. One thing that they highlight is the importance of acceptable industry standards of experimental design, so that things are being done consistently across multiple sites in terms of how sites are laid out, how large the sites are, how they're monitored, all of those kinds of things. And then the last thing I want to just highlight here, these are some of their recommendations in terms of if you're going to take this kind of approach, really important to think about simple protocols, modular experiments, standardized treatments, and this idea of what we do with the data that come out of these projects is something that many practitioners, in my experience, don't give enough time and attention to. And it's I think a really important aspect of trying to learn from these experiments while we're doing these restoration activities. So that's where I'll stop. If there are questions, I'm happy to answer them. You're also welcome to reach out to me via email if you've got questions, or if you want to see PDFs of any of the documents I talked about, or just bring ideas up. I'm happy to discuss those. Thank you.
We have a few questions. Has the grassland work been compared to U.S. Agricultural Research Service research, or Natural Resource Conservation Service and Soil Conservation District data and programs?
It hasn't been directly compared. You mean in terms, I'm not sure if the question is related to the effectiveness or to the actual products themselves. Some of the NRCS scientists have come out to our sites and have worked on the sites and interacted with us on them there. I guess one other thing I'll just say in that regard, when we have those experimental arrays where we have a whole bunch of treatments that are set up in relatively close proximity to each other, one of the really insightful aspects of that was the opportunity to go out there with lots of different people who have lots of different areas of expertise, and people can walk around and they can just look at these different treatments that have been applied in the same landscape, and they can bring their different areas of expertise to bear on that. So some people will look at a site and they'll say, well, I don't know, it doesn't really matter for their purposes what the treatments are, but this one looks like it's functioning really well because, maybe they're in charge of invasive control and they don't see as many invasives in that as they do in another. Or maybe they're a wildlife biologist and they're looking at it from the perspective of which of these is providing the best habitat for the organisms that they're thinking about. And so they can think about it sort of in a qualitative sense. And then once everybody has had a chance to look at it from their sort of viewpoint, then we can have a conversation about which of those treatments seems to meet the most of the objectives that people have identified. And so we've had some really fun conversations in the field in that context with folks from NRCS and from lots of different agencies.
Have you considered talking to farmers and ranchers as part of the analysis of the data collected?
Some of my colleagues have been working with ranchers. They've been doing some projects now that are starting to work directly with ranchers where they have livestock involved in grazing some of these prairies and looking at how that can be a management tool as well. We've shared the products of the analysis, I'd say, with them more than most of the farmers I've talked with haven't really been interested in the analysis itself. They've been interested in the products of the analysis, the conclusions that we've drawn from it. And we definitely shared that.
Do you have a repository or other means of centralizing project planning and information to facilitate coordination?
I think it would be, in the context of riparian restoration like this, I think there would be a need to establish that repository for it. The Nutrient Network, for example, has a repository that we've built for that information. And there would definitely be ways to leverage that for establishing a new repository for in this context.
While I like the idea of this approach, given the time frame needed to determine success when working with trees, I'm concerned about the time frame needed to develop results and recommendations. Any idea of how long it would take to develop improved riparian restoration recommendations?
I think you could get if you imagine however long it takes you normally, like if you imagine your kind of business as usual scenario and how long it takes you to determine whether that particular treatment has been successful or not. I think this sort of approach on an individual site in terms of the kind of scaling it for the needs of a particular site, that might double the overall time frame, but I would argue that it would be ultimately be more effective because you would be customizing the treatment so that what's being done on most of that site is what is most appropriate for that particular site. But I think you would also gain some pretty immediate benefits from that coordinated approach in terms of being able to assess initial survival, for example, across a wide range of sites and conditions that could be effective or could be helpful for improving restoration effectiveness pretty quickly after that.